Good morning, everyone. I am Dr. M. Mohammad Mahrub Raja, Assistant Professor, PG and Research Department of Microbiology, Jamal Muhammad College, Tiruchirappalli. I am going to discuss the chapter of immune responses. So, immunities is commonly defined as the resistant to infections. So, immunity is otherwise called free from burdens. So, ability of an organism to recognize and defend itself against a specific pathogen or antigens. Immune responses, it is a third line of defenses. It involves the production of antibodies and generation of specialized either T lymphocytes or B lymphocytes against specific antigens. Antigens are commonly called as the proteins or polysaccharide molecules from a pathogen or foreign organism that provoke a specific immune responses. So, immunities are broadly classified into innate immunity or genetic immunity. Another one is the acquired immunity. So, innate immunity is an immunity an organism is born with. It's a genetically determined one. It may be due to lack of receptors or other molecules. It's required for infection. Innate human immunity to gain distemper. So, immunity of mice to make poliovirus. Acquired immunities that an organism develop during his and her lifetimes. It is not genetically determined. It may be acquired naturally or artificially. So, the development of immunity to measles in response to infection or vaccinations. Types of acquired immunity, it's a naturally acquired immunity. So, naturally acquired immunities, it obtained in the course of daily life, that is naturally acquired active immunities and naturally acquired basic immunities. Naturally acquired active immunities, antigen or pathogen enter body naturally. Then body generate an immune responses to antigens. Immunity may be lifelong, example chickenpox or mumps virus, or temporary immunities, examples influenza or intestinal infection. Another naturally acquired basic immunities, antibodies pass from mother to fetus through placenta or breastfeeding. No immune responses to antigen. Immunity is usually short lived, that is a week to months. It may be protection until child immune system to develops. Then artificially acquired immunity is obtained receiving a vaccine or immune responses. There are two types, artificially acquired active immunities and artificially acquired passive immunities. In artificially acquired active immunities, antigens are introduced in vaccination, that means immunization. Body may generate an immune response to an antigens. Immunity can be lifelong or temporary. In lifelong immunity, example of oral polio vaccines, and temporary immunities, example for tetanus toxoid. Then artificial acquired passive immunities, its preformed antibodies are introduced into the body by injection. Examples of the artificially acquired passive immunity, snake antivenom injection from horses or rabbits. Immunity is short-lived, artificially acquired basic immunity is short-lived. Host immune system does not respond to an antigen. Serum, it's a fluid that remains after blood has clotted and cell have been removed. Antiserum is defined as the serum containing antibodies to a specific antigens. It's obtained from injecting an animal with antigens. Examples: horses, rabbit, and goat. Antigens: snake venom, botulism, or diphtheria toxin. Serology is defined as the study of reaction between antibodies and antigen. Gamma globulins defined as the fraction of serum that contain most of these antibodies. It contains most of these antibodies. Serum sickness, the disease caused by multiple injection of antiserum, immune responses to foreign protein. It may cause fever, kidney problem, and joint pain. Nowadays, it is very rare. Duality of immune system. So first one is there are two types: humoral mediated immunity. Another one is cell mediated immunity. In humoral mediated immunity involves the production of antibodies against foreign antigens. The antibodies are produced by a subset of lymphocyte is called as B cells. B cells that are stimulated will actively secrete antibodies and are called plasma cells. Antibodies are found in extracellular fluid, examples blood, plasma, lymph, mucus, etc., and the surface of B cells. It defends against bacteria, bacterial toxin, and viruses that circulate freely in body fluid before they enter the cell. It also causes certain reaction against a transplanted disease. Then cell mediated immunity, it involves specialized set of lymphocytes called T cells that recognize foreign antigen on the surface of the cell, organisms, or tissue. So types is helper T cells and cytotoxic T cell. T cell regulate proliferation and activity of other cells of the immune system. B cells, macrophages, neutrophils also comes under the cell mediated immunity. It defends against bacteria and viruses that are inside the host cell and are inaccessible to antibodies. Fungi, protozoa, and helminthes, the cell mediated immunities act against fungi, protozoa, and helminthes, cancer cells, and transplanted tissue. Then antigens. So antigens, most of the antigens are proteins or large polysaccharide from a foreign organisms. In microbes, capsules, cell wall, toxin, viral capsid, flagella act as antigen. In non-microbes, pollen, egg white, red blood cell, serum protein, and surface molecules from transplanted tissue acting as non-microbes antigens. So lipids and nucleic acids are only antigenic when combined with proteins or polysaccharide. The molecular weight of the antigen 10,000 or higher is a great antigen or good antigens and able to make the immunogenics. Haptens are small foreign molecule that is not antigenic. It must be coupled to carrier molecule to be antigenics. Once antibodies are formed, they will recognize haptens. Then epitopes, antigens molecules are larger or very longer length. Only small part of an antigens that interact with antibodies. The small part that may be act as epitopes. Any given antigen may have several epitopes. So each epitope is recognized by a different antibody. Each epitope is recognized by different antibodies. Then antibodies are a glycoprotein that recognize and bind to a particular antigen with a very high specificity. They made in response to exposure to the antigens. One virus or microbes may have several antigenic determinant site to which different antibodies may bind. Each antibody has at least two identical sites that bind antigens, that is called antigen binding site. Valence of an antibodies, number of antigen binding site, most are bivalent. It belongs to a group of serum protein called immunoglobulins. Antibody structures are that is a monomer, a flexible Y-shaped molecule with 4 polypeptide chains, 2 identical light chains, another 2 identical heavy chains. In variable region, 2 section at the end of the Y arms, it contains the antigen binding site. It is called as Fab. Identical on the same antibody but vary from one antibody to another. In constant region, stem of monomer and the lower parts of Y arms. In FC regions, stem of monomer only. It's important because they can bind to complement or cells. Then classes of immunoglobulins. So first one is the IgG, immunoglobulin G. The structure of IgG is monomer. Percentage of serum antibodies found in 80%. Locations of IgG molecule in blood, lymph and intestines. Half-life in serum only 23 days. IgG's complement fixations done by IgG molecules. Then placental transfer is done by IgG immunoglobulins. Known function, biological functions of the immunoglobulin, it enhances the phagocytosis, neutralize the toxin and viruses, protect fetus and a newborn. Then IgM is a pentamer structure, percentage of serum antibodies in 5 to 10%. IgM is located in blood, lymph, B cell surfaces. Half-life in serum so only 5 days. IgM then complement fixations. There is no placental transfer. The functions, biological functions of IgM is first, it may be a first antibodies produced during an infection. It's effective against microbes and agglutinating antigens. Then IgA, the dimer structure, percentage of serum antibody 10 to 15%. Location, secretion like tears, saliva, intestine, milk, blood and lymph. Half-life in serum 6 days. The function of complement fixation is nil, placenta transfer also nil. Biological function, localized protections of mucosal surface, provides immunity to infant digestive tract. In IgD, immunoglobulin D, the structure is monomer. The percentage of serum antibody is 0.2%. It located in B cell surface, blood and lymph. Half-life in IgD is only 3 days. The biological function is unknown. In serum and B cell surface, it initiates immune responses. IgE, the structure is monomer. Percentage of serum antibody is 0.002%, located in found to mast cell and basophil throughout body and blood. Half-life in serum only 2 days. The biological function is allergic reaction, possibly lysis of worms. I think all are understanding the structures of antigen, the structure and classification of immunoglobulin and able to make the immune responses. So thank you everyone.